So 6-carbon compound is broken down finally into 2-carbon compound called pyruvic acid. Okay, so this pyruvic acid will enter the new mitochondria from the cytoplasm, and it will perform two main reactions. So number 2, and then from the number 2, number 3. Okay. So number 2 is the one which is called the citric acid cycle. It has many names. Other name is Krebs cycle. Citric acid cycle. And citric acid cycle is a series of reactions, which actually I showed you last Thursday. It is actually performing again a series of reactions, but it is going over and over in a cycle. And that process is also make ATP. Okay, so we have ATP made here. ATP molecule, the energy molecule, sorry, ATP. We also make ATP in citric acid. This is I, not you, I. It makes ATP also. But this is again not the main amount. It is also small amount. And from the citric acid cycle, we have some compound produced. And that is actually, we'll see here in this pages, NAD and FAD. These are it's called cofactors. Those are the precursor or compound which will go to participate in number 3 reactions. So number 3 is called the electrons transport chain, ETC. So you see number 1, glycolysis taking place outside the mitochondria in the cytoplasm. Number 2 is the one which is taking place inside the mitochondria. That is citric acid cycle, which is a cyclic reactions, many reactions going over and over. And ETC is the 1/3 one, which is also taking place in the mitochondria using the materials which are produced in the second step or second reactions, which is citric acid cycle. ETC is the one which produced the major amount of ATPs. Okay, so if we start with one glucose molecule, okay, for example, then the pyruvic, the first one, the glycolysis will produce 2 ATP. The citric acid cycle will also produce 2 ATP, and 34 ATPs will be produced in the ETC cycle or ETC reactions. So that is the major one. So usually we get 36 to 38 depending on some metabolic reactions. So one glucose molecule, which is actually the ultimate product of the carbohydrate digestion, will give rise total 36 to 38 molecules of ATP. So these things you need to remember, especially the 3 steps and the number of ATPs which are produced. And these ATPs which are ultimately produced, only 40% are used for the cellular activities, and these are the cellular activities. 60% are not used. So we always make more ATP than our cell is requiring, or the whole body is requiring. And that is had advantages that we are living. Otherwise, if we have scratch, we have not surplus, but opposite, you do not have enough ATP, then we would not survive. And this 60% ATP actually are lost through the body as a heat energy. That's why our body is always warm. You touch the body compared to atmosphere, it is always warm. Okay, now we will see these two reactions, especially the citric acid reaction and the ETS, electron transport chain reactions. And then we'll come back to the glycolysis later. Okay, so citric acid cycles actually, as I said, it is actually start with the 2 carbon molecules. And these 2 carbon molecules actually originally came from 6 carbon of glucose. And when it enters the mitochondria, it turns into acetate. And then it binds with another compound called acetyl, actually coenzyme A. So acetate entering the, it is actually converted from the pyruvic acid, which I said that it is the ultimate product of the glycolysis. So when it enters the mitochondria, it is converted into acetic acid or acetate, and it combines there with the coenzyme A, and the ultimate product will be acetyl-CoA. This is the first, it's called the reactant that is entering the cyclic reactions of the citric acid cycle. So let us see what is how the citric acid cycle looks like. Citric acid cycle, it is just it's a summary of this. And here we also produce, citric acid cycle also produce the carbon dioxide. So in the respiratory system, we said that the cell also produce carbon dioxide gas as a byproduct, and it has to go out. This is the area when we make the energy, we also make carbon dioxide as a byproduct. And here also we use the oxygen. So one thing you should know that when the reaction is taking place inside the mitochondria, here oxygen is used in both ETS and citric acid cycle. So these two reactions are combinedly known as the oxidative respiration, where the oxygen is used. But before that, the number one, the glycolysis, which is taking place outside the mitochondria, no oxygen is required. So that reactions together is called the anaerobic respirations, because no oxygen is used there. So these two products from the citric acid cycle, they are byproduct, but they are not like carbon dioxide. They are not unwanted. They are wanted. They are very useful compounds or molecules, and they enter the third reactions where we get ultimately the 34 molecules of oxygen, ATP. Okay, now we'll see the reactions of the citric acid cycle. So here, this is actually showing you not everything, but you have an idea how these reactions are taking place in a cyclic form. So you see the first material, which is pyruvic acid, which is not showing here, but I'll show you later. It enters the mitochondria and then it is converted into acetyl-CoA. First, it will convert into acetic acid or acetate. Okay, acetic acid is actually a good example is vinegar. Vinegar is the acetic acid. So this acetic acid is combined with the coenzyme, which is actually coming out inside also from cytoplasm. And then it makes the acetyl-CoA. This is the first reactant entering the cycle, and it enters, and then it is taking place. It is actually reacting with many other enzymes. So in each step, we have one or two or more enzymes. That's why it is not 100% showing you what is actually happening. If you have taken the biochemistry course, then each of the steps you have to memorize, and you have to know which enzyme is coming here and what is being made from the previous compound. So we had acetyl-CoA that is converted into citric acid cycle. Okay. So now citric acid. That's why the whole reaction is called citric acid cycle. The citric acid is a 6-carbon molecule. So we started with the 2, then we have the 4 here, and then we have the 6 here. So we actually making the citric acid cycle with the help of enzymes. Now, when the next step from the citric acid cycle, we also lose 1 carbon, and we make 5-carbon molecule, and it has a name. It's called glyceraldehyde. But for you, since you are not biochemistry student, just have some idea. Where is that 1 carbon gone? 1 carbon is used to make the carbon dioxide gas. So this is the first source of carbon dioxide produced in the cell. And then on the other side, we have NAD. Okay, NAD is there, and it is actually energized. It gets hydrogen, and then it is made NADH, usually NADH2. So hydrogen is added to it, and this molecule NAD become now an energized molecules, and it is going towards the third experiment, third reaction, number 3. This is number 2. This is number 3 here, and then you will see how that reaction is taking place here. So NADH2 is going there, and then in that next stage from 5 carbon, it is getting into here. So in between we have some other steps which is not showing here. So we get next one is showing here 4 carbon compound. So 1 carbon is again lost from here, and which is again the second molecule of carbon dioxide is produced. And here also we have NAD is turned into NADH2. And then the very important thing is the ATP produced in the molecule, energy molecule produced in the citric acid cycle, it is taking place in this stage. Okay, and this ATP is produced by making the GTP into GDP, and this 1 phosphate is added to ADP. So actually ATPs are always made from ADP, and ADP are made from AMP. That is a general rule of the biochemistry of this metabolic reactions. So AMP means 1 phosphate, ADP means 2 phosphate diphosphate, and ATP means triphosphate. So it has 3 phosphate. So this is 1 molecule of ATP we have for each cycle. And then after the 4th carbon compound is produced, it will turn into 4 carbon molecule again, but it's different. The names are different. This is not the same as this one. By this time, we produce FAD. This is another compound like NAD. So FAD will be now, you see these reactions there, and then also another 2 NAD will turn into NADH2. These 2 compound, also they are called cofactor, they are also coming back to join with the previous ATP, and then they will be entering the third reactions, which is ETS. So this way we know, and after the 4 carbon compound, it is converted to acetyl-CoA, acetate and acetyl-CoA again. So this way the circle is going on and on. If for each each molecule of carbon, each molecule of glucose, we have one cycle, and then here we have one ATP produced. Okay, next, we will see what is going on in the third sets of reactions, which is ETS. So ETS is called because it is an abbreviation of electron transport system. Electrons, it is sometimes called electron transport chain also, because it's a chain of reactions taking place. And here, the energy, which I just made, not ATP, the ATP is required. When ATP has to be made, you need energized compound, which are NAD, NADH, or those compounds. Those are carrying electrons, and that's why the electrons are energized molecule. Compound, sorry, not compound, not entity, we can say, subatomic particles. Those electrons are carried by the compounds involved in the this whole reaction. That's why it's called the electron transport chain. So here, this is called also, this is the part of the oxidative respiration. And this part is called oxidative phosphorylations. That means we will use oxygen here, and then also the making of the ATP is also called the phosphorylation, means adding of phosphate. So ATP is made from ADP. So one ADP is added with it one phosphate, and that's why it converted into ATP. So this is called the phosphorylations. So oxidative phosphorylation, both reactions are taking place in this this stage of the energy productions, and they also need coenzymes. Coenzymes, now we will see this NADH2, FADH2, those are called coenzymes. They also use the oxygen, and finally we get the ATP. But in addition to that, there are a series of enzymes involved. So all of the reactions, what you eat, what you see taking place, either metabolic anabolism or catabolism, they must use the enzymes. Otherwise, it cannot go through in our cells. So 95% of ATP are produced in this process. And the rest of the 5% we produce either by glycolysis or the citric acid cycle. Okay, so now what are the enzymes which are involved in the ETS cycle? ETS cycle involved the cytochromes. Chromes, that means there are many of them. They're called a cytochrome A, B, C, D, and each of them are like working like a relay race. So one will get the electrons and it will transfer to the other one. And when it transfers to the other one, it goes to higher level of energy. And this way, a series of cytochromes are involved to transfer the electrons and ultimately get into the higher energy level. And then we get the formation of ATP. And here, one of the enzymes which is ultimately used, cytochromes are enzymes, ATP, and this enzyme is not the cytochrome. This is the other, a different kind of enzymes called ATP synthase. This synthase means which can synthesize molecules. So this ATP synthase is an enzyme which will ultimately produce the ATP molecules. And here you can see the number 3, which we have two here. We have one here. One is glycolysis, 2 is citric acid cycle. Now we have the number 3, which is showing here what is happening. It is happening it is in the membrane of the mitochondria. And where we have this series of cytochrome. This is one, the first cytochrome. This is the second cytochrome, and they have actually named cytochrome A, B, C, and others, not A, B, C, D, E, F, some others also. So here the cytochromes are getting the electrons with the help of the NADH and FADH, which came from the citric acid cycle, and this energy electrons are transferring. You see that they're transferring, and then each time it is transferring, it goes into the higher level of energy. And finally, with the help of this one, this is the ATP synthase. Okay, ATP synthase enzyme is here, ATP synthase, which is converting ATP to ADP to ATP. A very important a byproduct of this reaction is the productions of water. So our cells also producing water all the time. And where this water is coming from? From these reactions, the ETS reactions, right? So this is very simple way, but otherwise it is so complicated for complicated reactions taking place. That's why I was saying that if you were a biochemistry class, then you have to know all of them. So one thing here, you see that this is NAD and FAD are converted back, because here they were NADH2 or FADH2. NADH2 or FADH2. This H2 or a hydrogen, these are the one which is carrying the electrons. And also you see the H2H is released, proton and electrons. So this proton is going all the way to produce the ATP, to produce the ATP at the end. All right, I know these are little bit difficult for you to grab, but I explained as much as simple way I could. If you have any questions, you can ask me at any time. Okay. So mostly the difficult part is over now. Now going back to the information we had talked about, that the cell has the nutrient pool, where we have all the product of the digestive system, digestions, and also the product of the catabolic reactions of the cell are they are deposited, and they are working like a source of material, source of nutrient materials. That's it's called the pool. And then it is the one from where the materials are taking place. So now we'll see one of the important aspects of using these materials from the nutrient pool. So the reactions I have mentioned before from the glucose, and glucose is the always use first. So now if you do not have glucose, what will happen? First, it will be using the fatty acids, but fatty acids, that means the source of the fat or the lipid, and it will also make the same 3-carbon compound which we had made by the glucose. Okay, that 3-carbon compound which will enter the mitochondria before it turns into the 2-carbon acid. The acetic acid or acetate will enter here, but this 3-carbon compound is the pyruvic acid. So glucose makes the pyruvic acid. The last of the glycolysis reactions, same will be made by the fatty acid and amino acid also. So ultimately, the reactions taking place in the mitochondria are same for all these 3 compounds or source of energy, like amino acid, fatty acid, and glucose. But in the only thing difference is the reactions. These reactions from amino acid to 3-carbon and fatty acid to 3-carbon and glucose to 3-carbons. These 3 are different from each other, but ultimately we need the compound which will be used for the production of ATP. Okay, now when we talk about the nutrient pools, so where exactly in the cell or in the body we have these reserves? So mostly the cells of the livers and are the one which is a source of the glucose, and this is called the glycogen. So glycogen, again, I told you before that it is the polysaccharide, or we have the reserve carbohydrate in the body, and that comes from glucose. So when we have surplus of glucose, okay, after making the energy, we have still huge, because we eat lots of carbohydrate beyond our requirement of the body. This extra glucoses actually, many glucoses, they combine again and make the glycogen. And this glycogen is stored in the liver cells mostly, but we also have in the muscle cells, the skeletal muscle cells. And this liver also store triglycerides. Okay, so triglycerides are like the polysaccharide. It is the fatty acids together. So triglyceride is the lipid which will be we need to be reserved in the body, and we can find it in the liver cells. So when we need to make energy, the glycogen has to be breakdown, and this breakdown of glycogen is called glycogenolysis. Glycogenolysis. And this glycogenolysis is the one by which we can break down and get the glucose. And this glucose will be subjected to the reactions to produce the energy. And triglyceride will be subjected to the lipolysis. Lipolysis is the process by which the lipid is broken. And you remember that you must know that all these reactions needed enzymes to perform. So lipolysis. Mostly the lipolysis is done by the lipase enzymes. And then we have a fat molecule, adipose tissues. Those are very much, we can see without a microscope with a yellow color. Now I'm not talking about the human body, but any other, like if you have a chicken and then you open the skin, you will see yellow color materials. Those are adipocytes. Adipose tissue is the tissue. Adipocyte is the cell of the fatty cells. They store triglyceride, not the glycogen. And then the fatty acids can be released from the lipolysis of the adipocytes, of the triglycerides. And the last source of our nutrient pool is skeletal muscle. And skeletal muscle, as I said, glycogen is the source there. And also the proteins, like the skeletal muscles contains the proteins, the muscle protein. The two famous or main proteins of the muscles are called the actin and myosin. They start now break down and give rise to amino acids. And these amino acids, if you go back to this previous slides, here you can get the amino acid. They will convert into the 3 carbon and 2 carbon, and finally get into the mitochondria, and then perform these 2 reactions, same as we saw for the glucose. So now you see, generally, when we have enough glucose or glycogen, then we do not worry about not getting more food. Once in a while, we get hungry and we go starved. That time, our body actually break down the glycogen and get glucose. When the glycogen, for example, you are not eating for days after days, okay, and especially carbohydrate, you do not eat carbohydrate. Then what happens? The cell will break down the fats from the adipose tissues as well as from the liver, and then we'll burn lots of fats, and we get actually skinny, or we lose our weight. That's a good thing, but you be careful that we do not do these things very abruptly, then we can have other problems. And then the last thing is like dangerous or the deleterious for our body when we do not have any more fat, triglyceride to convert it into the energy. And then what we'll have, our proteins in the muscles are broken down. That means what? The most muscles are broken down. So extreme starvations, which is actually a very serious situation, can turn people not only skinny but very fragile. Their bodies, skeletal muscles are torn, actually they are damaged. This this is called malnutrition. One of the malnutrition of this type of situation is called the kwashiorkor. And you can see people who are in famine hit countries in Africa, how skinny they become. And so their body is completely devoid of not only fat, and started breaking down into the breaking down of the protein. Right. Okay, here this is a summary of summary of what I just mentioned, that where we actually summary of the nutrient pools, use of the nutrient pools for the making the energy. So if we start from the blood, okay, here, because blood is the ultimate source of everything. It carries from one place to another place. So we have these are the cells. This is the cells where we get the triglycerides and glycogen, and this is, we can say this is the liver cells. Sorry, yes, this is the liver cells, which is called the hepatocyte actually. And then it is breaking the glycogen. When glycogen break down, then it is actually making the glucose, and this glucose is used for the energy, and this energy is used for like cell divisions, absorb and catabolize the glucose. Then these are the adipose tissues or the fat tissues, where mostly we get the source as a triglycerides, and triglycerides will be broken down, and also triglyceride will be made from excess of the fat by anabolism and stored there. And then you see I'll come back to that one later. Now this is the intestine. It is talking about the metabolic reservoir from there, because from there ultimately after digestion, all the amino acids, all the fatty acids, all the glucose, they are coming and go to the blood. From blood we take to the cells. So this process from getting nutrient materials from the intestine to the blood is called, if you remember, absorption. We learn it in the digestion subject. And then we do not get anything from the nerve cells. Nerve cells are the one who needs always supply of the nutrient. So especially during the starvation, the tissue shifts to fatty acid or amino acid that I just told you, but mostly the glucose from the nutrient pool should be transferred. And the neuron cells, this is the nerve cells, they do not have capacity like this one, and this one to store the nutrient pool or store the any nutrient materials that could be used for its own purposes or for others. So that's why these cells are very much vulnerable. And for them, we have other cells. Now, those are also nerve cells. There are 6 types of cells. They are nursing them. These are called glial cells. Okay, the last one is the muscles. So this is the skeletal muscle showing, mostly the skeletal muscle, not the other two muscles. They are the source of the energy, because they get deposited this glycogen in their glycogen. This is present here, and this will be broken down. As I said, that in extreme conditions, the protein also broken down here, and then we get the amino acids, and amino acids goes there. All right, so this is also same thing I just discussed before. So let us finish this part here. Okay, now something I have actually discussed in my writings on the board. So that is actually showing here, and this is that the terminologies are very similar to each other, but you have to know the differences. So there are the dynamics. Dynamics means both breakdown and synthesis. There are three ways which I have mentioned. One is the glucose first. Glucose is synthesized, glucose is used for the energy, and glucose is also being broken down, and the glucose is also used for the energy. So glucose, I am coming back to the fatty acid after I finish it. So glucose, when we break down the source of glucose in the body, which is glycogen, this breakdown of glycogen is called the glycogenolysis, which should be showing here, glycogenolysis. So glycogenolysis will be broken down and we get glucose. And when we have many glucose together make the glycogen, this is called glycogenesis. So see, they're very similar synonyms or nomenclature, but differences is the last part, glycogenesis. Genesis means synthesis. Glycogenolysis, glycogen, this is the one part, and lysis is the breakdown. And then we have also talked about, if you go back here, you see that glucose, when we do not have glucose, but we make ATP from the source of amino acid and fatty acid. Okay, so these fatty acid amino acids, they are not the carbohydrate source. One is protein source, one is the fatty acid source. So when we make glucose, so then our energy molecule from the non-carbohydrate source, this is called gluconeogenesis. Gluconeogenesis. Gluco means glucose. Neo means something new, not traditional thing. Neo, something naive. So neo, and genesis means production or synthesis. So gluconeogenesis is the making of glucose from the non-carbohydrate source. As when we do not have glucose or glycogen, then we have to make energy. So that's why we produce glucose from the fat and the protein or amino acids. Those are called gluconeogenesis. Okay, and here we do not produce glucose. Remember, glucose is glucose. So we produce something which actually we make from the glucose when we make the energy from the source glucose or glycogen sources. We make carbon acid chain compound, smaller carbon chain compound, like the 3-carbon or 2-carbon compounds. So those are the, but we name it gluconeogenesis because it is a carbon chain, which is glucose, but not glucose. Okay. And the last one is another terminologies, which is the number one reactions. So if you remember that the first reactions for in general, what happens? We have glucose. Glucose is converted into the pyruvic acid, and this is happening outside the mitochondria without the help of oxygen. So that process is called the glycolysis. Okay, so now we have this 3-carbon chain compound. That is actually the one which is ultimately we get it to enter the citric acid cycle. This is also made from the gluconeogenesis. So now you have 4 terminologies very similar to each other: glycogenesis, glycogenolysis, gluconeogenesis, and glycolysis. Okay. So this you have to know the differences between these processes. All right, so I have discussed about the fatty acid and amino acids, so I don't want to go back again. So this is the one which is all together. This is the now synopsis of the entire process of making energy with the glucose source, glycogen, protein source from amino acids, and fat source, the fatty acids. And they all ultimately has to make this compound, which is 3-carbon compound. So 3-carbon compound, and then it will be turn into 2-carbon and then enter the citric acid cycle and then electron transfer cell. So this is like synopsis. I don't have to describe again. What you see also here, the oxygen is required, and this is required for the ETS, and carbon dioxide is produced, and water is also produced. But mainly we are looking for the production of ATP. Okay, so now we have some easy part here, and this easy part is just description, but you don't think critically, just read it and you will understand. So this is the what we call the understanding of the digestive process in terms of metabolic reactions or metabolism. Okay, so here we have different parts, different places of the body, especially of the digestive system. What is happening in terms of energy productions ultimately? So oral cavity is the one where we have the mechanical process by the use of teeth and tongue, and here we have digestion of the carbohydrate and lipid. If you remember, the carbohydrate is digested partially by the amylase, which is coming from the saliva, and saliva also has salivary lipase, which also digest not completely the lipid materials. Stomach is the one where mainly the digestion is taking place with the help of acid, but it is not the only one. We have enzymatic digestion of the protein there, and then in the duodenum of the small intestine, we have many catalytic enzymes. That means it is the one which breaks down most of the organic materials which are not digested in the or incompletely digested in the stomach. And after that, the jejunum, ileum are the place. They are also they have some finishing of the digestion, but mainly it is the place where the nutrients are absorbed. After digestion completed, the nutrients are going to the blood, and then it is either stored or it is processed in the liver. If it is used, then it goes to the pool in the cell, and then make the energy. And in the large intestine, we do not do any digestions. Only water, which we drink a lot every day with, or also we have with the food, they go back to the blood. Okay. Otherwise, if you lose all the water, then blood volume will decrease day by day. And some nutrients and vitamins which are produced by the bacteria are actually made here, and large intestines also is responsible for accumulating and excretion or elimination of the feces. So now we will go some details about the different different food materials. So we start with the carbohydrate. So carbohydrate is the one which is first digested in the mouth with the help of salivary amylase. Salivary is the saliva coming from the salivary glands, and that digests the carbohydrate into disaccharide and trisaccharide. You see that usually when we eat bread and rice, it contains the polysaccharide, which is called the starch. Remember, starch, just give you some TARCH. And these are the three examples of polysaccharide, carbohydrate. Starch is the one which we eat. Glycogen is the one which we make it in the body as a reserve of the carbohydrate. Cellulose is the polysaccharide which we do not have capacity to digest, but we eat it with the when we eat many things like fruits. The scale of the fruit is the cellulose, but it is digested by the bacteria. So if I give you the example and ask you to give the example of polysaccharides and what are they actually, what are the sources, you should know. Then in the duodenum, the complete digestion of the carbohydrate takes place by the enzyme called alpha amylase from the pancreas. So this amylase from the pancreas and this amylase from the salivary gland, they are not exactly the same. This cannot digest into monosaccharide. They make the like disaccharide or trisaccharide. Now, in the jejunum, we have maltase. So if the disaccharide is coming from the mouth, it will be now turned into monosaccharides. Okay. Then we have sucrase. Sucrase is the one which will digest the sucrose. Sucrose is the sugar we eat every day. Then lactase is an enzyme. If we drink lots of milk, then we have lactose disaccharide. Then it will turn or break down into monosaccharides. And in the largest intestine, remaining indigestible carbohydrate, I just mentioned, cellulose, are food source for colonic bacteria. So bacteria has an enzyme in the small intestine. This enzyme is called the cellulase. That cellulase will be used to digest the cellulose. This one. So now in the about the glycogen. Glycogen, I said that this is the stored carbohydrate in human body. In skeletal muscle, they are stored in the liver also. And this glycogen will be break down by glycogenolysis. to make the glucose. And then this glucose will be used, and then it will be converted to pyruvic molecules. Pyruvic acid, that I said that is the last compound of the glycolysis, and they are three carbon compound. So if you break down glucose is 6 carbon, and if you break down into half and half, then we'll have 2 pyruvic acid, 3 and 3. And that will be producing the energy in the process of glycolysis. We will have only 2 ATPs there. Pyruvate used by the mitochondria uses carbon dioxide and generates 34 ATP ultimately. 34 and 2, 36 also, if you add the ATP of the citric acid. So here the sketch, it is showing. So if you have glucose 6 carbon, then it will be converted into it will enter the cell, okay, and it will be in the cell, it will make this one, and this is the number 1 reactions called glycolysis. And then you have the ATP productions. Here also actually 2 ATPs are produced, and then pyruvic acid will be adding with the coenzyme A and make the acetyl-CoA, because this 3 carbon compound pyruvate will turn into the acetic acid. So I don't want to go again the same thing over and over, but I have talked about this. Since this is the glycolysis, so this process here taking place is called glycolysis. This is anaerobic respiration, and here ultimately the pyruvic acid, which is the 3 carbon compound, are produced from glucose, which is 6 carbon compound, and it is taking place in the cytoplasm, which is also known as cytosol. Then it produces 2 ATPs. It also produces hydrogen atoms that are transferred to NAD to mitochondria. So glycolysis process are described here with the descriptions of what we get. So total, we get 2 ATPs from one. So when we talk about this, we are talking about 1 carbon, 1 glucose molecule. So we have millions of glucose taking place during the process. But if we want to know the exact numbers, then we have to consider first how many ATPs are coming from 1 molecule, and then you can multiply with the number of glucose we eat or we produce or we use. So there will be millions or billions or trillions of ATP molecules we produce, which are 60% above our requirement by the cells. At the same time, in the glycolysis, we have the water molecule released. And here, this is a very important slides or information regarding the glycolysis, which I have not talked about that before. So you don't worry about that because it is too many things here. But if you were biochemistry students, you need definitely required to memorize it, all the steps. So you see, what actually I want to tell you, this is first thing, the glycolysis is taking place in the cytosol, in the cytoplasm, not in the mitochondria. So what is the beginning? The beginning is the glucose, which is 6 carbon molecules. Okay, it enters the cell, and glucose comes from the carbohydrate digestions. Everybody know it very well. When it enters the glucose in the cell, normally it happens. In diabetes, this glucose is difficult to enter. Okay, and that's why we have a high level of glucose in the blood. So it is in the blood actually. This is coming from through the blood. We know that. Okay, now insulin comes here and binds with the, so this is the protein or enzyme, which is membrane bound. And then insulin, this is just, I'm giving you some different information. So insulin is also in the blood. It comes and binds here. When it binds here, then the glucose is easily go inside the cell, and our glucose blood level of glucose is reduced. So now when the glucose enters, it is using 1 ATP. So this reactions is consuming ATP also. It is making ATP, but it is also consuming ATP. So 1 molecule of ATP is used here, and this it is turned into the ADP because a phosphate is removed and this phosphate is added to the glucose. So now it turns into glucose 6-phosphate. And now the second step, we also use another ATP. See, and then you see that this glucose molecule has another phosphate. Now it has 2 phosphate in 1 glucose molecule, and this is called fructose 1,6-bisphosphate. This is fructose. It is now converting into fructose. So also remember, which is not showing here, for each step we have different enzymes. In biochemistry course, we have to know and memorize also this name of these enzymes. And next step, what happened next, step number 3, this is divided into 2, half and half. So we get, you see that either we get both. 1 molecule is called, with the 3 carbon, is called dihydroxyacetone phosphate. And the other molecule is called glyceraldehyde 3-phosphate. This is one. Glyceraldehyde 3-phosphate. Now what happens, this dihydroxyacetone phosphate comes back, is converted into the glyceraldehyde 3-phosphate. It is very unstable. So it is converted, and now we have 2 molecules of glyceraldehyde 3-phosphate. We'll write it like this, G3P. Then we are getting NAD from the mitochondria, and it turns into NADH, and it goes to the mitochondria again. And here, the next step is this compound, 1,3-bisphosphoglycerate, because it is using 1 phosphate here, not the ATP, but the phosphate only. And then when this molecule is converting, it makes 1 ATP here. Now you have the 6th step. In the 7th step, which is the last step of the glycolysis. This whole process is called the glycolysis, remember. It said here, steps in the glycolysis. Last step, we are producing another ATP molecule, actually 2 ATP molecules. So the main idea or main information I want to give you, that in the whole glycolysis process, we produce actually how many? You see that? 4 ATP. 4 ATPs. But in the whole process, we also used or consumed 2 ATPs. So here it is said, step 1 to 2, we use 1 and 2, we use 2 ATP. That's minus. Step 5 and 6, this is 5, this is 6, we use 2 ATP too. So total, we have used we made 4 ATPs, but we used up or consumed 2 ATPs. So this is the net gain. That's why we say that in the glycolysis, we produce 2 ATP molecule from 1 glucose molecule. But ultimately, actually we produce 4, but we used 2. So the net gain is 2 ATPs. And 4 ATP. So this is the summary of ATP production in the aerobic ATP. So we don't have to. So there is the information here. It says why glucose is the primary source. You remember I told you that the cell will look for glucose to make the energy. Then if this glucose is not there, then it is use the fat and then protein. So why the cell should use the glucose? Because there are some reasons. The first reason is glucose is the small molecules and soluble in water highly. Fat is not soluble in water, and fat is big molecule. What protein is soluble, or protein is also big molecule. So cell always try to use small molecules because big molecules you need to use more energy. Then the glycolysis process is the one which provides a small amount of ATP anaerobically. So that means you do not have to use the oxygen here. And then you are, number 3 is glucose can be efficiently stored as glycogen, which forms compact insoluble granules. Okay. This is actually another point why the glucose should be used, because we want to get rid of glycogen. Why? Because glycogen is big molecules and glycogen is insoluble granules. So these are not favorable for the cells. So that's why, and more you can break down the glycogen, more is the cells are more happy. Then we have the last reason is the breakdown of glycogen, which is known as glycogenolysis, occurs very quickly and involves only a single enzymatic step. So this is also another advantage the cell will take place, that to convert the glycogen into glucose, it is quick and it is one-step process. One enzyme is involved. If you want to make, you cannot make the glucose, but you make the compound, which is ultimately 3-carbon compound, which is pyruvic acid, from the fat and from the protein. It is many steps. There's many additional steps. So that's why the cell will not go for the expensive one, but go for the cheap and fast one. Use the glucose, and then if glucose is not available, then it will go for the expensive processes. So I have 5, 6, 7 minutes. So let me see. Just let me finish the lipid part. So the carbohydrate part is gone. So lipid is the one which started being digested in the mouth with the help of lipase. This is called lingual lipase, means it is coming from the mouth, and it is mouth means it is coming from the saliva. Okay, but in the stomach, the lingual lipase continue to function. It is still there and it is functioning, but only excess surface of the lipid that have formed. Now, to digest the lipid molecules, you need, or complete digestion, you need to convert the lipid molecules into a form called the emulsified, or this process is called the emulsification. Emulsification, you can see here, so this is the lipids, and then with the help of bile, it makes this kind of round shape. This is called the micelle. But a micelle is the form of emulsifications. And then emulsification is the one which is favorable for the enzyme pancreatic lipase to digest the triglyceride or lipid. Okay. So in the lipid, we have new triglyceride, which is called the chylomicrons, chylomicrons are the large molecules of lipid. Okay, so now you can see the distribution of the chylomicrons and other. So we have lipoprotein particles like lipids. They do not. They are not soluble in water, or especially in the blood. So what happens? These lipids they conjugate. They make bond with proteins. So that's why they are called lipoprotein. So we have triglyceride, 95, more than 90%, mostly present. Phospholipids are 6 to 12%. Cholesterol, it is about 1 to 3%, and proteins is 1 to 3% in the chylomicrons. Chylomicrons are large, very large molecules. So this is the combination of different kinds of lipids. It's not only one. And they transport dietary lipids from the intestine to the location of the body. So I think I'll not be able to finish because there are a number of lipids we have to discuss. There are LDL, HDL, and also it's called the good cholesterol, bad cholesterol. So we have to spend some time before we start the protein. So when we finish this lipid digestion and protein digestions, that will be the end of this chapter. I will not go further, and you will have your quiz on this chapter. So most likely, not most likely, we'll be able to finish on Thursday, and we'll start a new chapter. And then you will have your first quiz, lecture quiz on this chapter. So which day? Probably it will be Tuesday, next Tuesday.
May I ask you a question, please?
Don't ask me about the please don't ask me about the review.
No, it's about tomorrow. It's about tomorrow.
Class tomorrow, what happened?
We have a lab exam on the respiratory, right?
Yes, in the lab, yeah.
So it's only going to be respiratory. It's going to be respiratory and digestive.
No. I did not say that digestive.
Just respiratory.
Two lab. Digestive, we have not started. The lab is not started.
Yes.
We'll start tomorrow.
It's going to be 8 a.m., Professor?
In the class, yeah, in the beginning. Please come on time tomorrow.
And you said on Tuesday, next Tuesday, we're having a test on this, on digestion.
Yes, quiz, 10 question probably.
Tuesday, but Tuesday is a lab. Tuesday is an online class.
Yes, it's a lecture quiz, I believe.
Okay, thank you.
Noor Muhammad, Noor, what you are talking about? Your class is online. Your quiz will be online.
Yeah, but this is the first time this is the first time you've given us a quiz of 10 questions on the online. That's why I was a little shocked.
Shocked? Why?
Usually we have
because usually your tests are like 50 questions. Why?
Yeah.
That is exam. I said that's exam. The exam and quiz are different. Quiz should be always short.
Okay.
So you want 50 questions, I can give you. So if you...
No, it's okay. We love your 10 questions.
So you said you're shocked. You should be happy.
Professor.
Yeah.
I want to make sure that I pass this test tomorrow, and I know the other classmates as well. Can you give us as much advice as possible to prepare for this test tomorrow? Like, is it all images? Is it multiple choice? Fill in?
Yes, please.
You will have images and you will have questions also. So try to. In the lab I explained what questions you should or what we should study.
Okay.
Study all the structures of the respiratory systems, like identify alveoli. So I to give you the picture and identify the terminal bronchiole, this kind of things. And for the physiological lab, mostly what we did experiments. We did experiments on the respirometer to determine the lungs capacity, lungs volume. So you have to know those formulas. We made some experiment with the acidity and alkalinity of the lime. So what you did actually and what you got the result of, those are the things. We also have the balloon and that this is called Bell-Jar reactions. And what happened if you pull and push the diaphragm. So I gave you some questionnaire, remember?
Yeah, so you said it was because the muscle pulls harder, right? Because you're using more muscle, that's why more air.
Nothing to do what I'm asking. I remember when you did the experiments in the lab, at the same time you answered some questions. Okay.
The table.
The questions still you have it in your slides if you go back to the bright space. So those questions will also be there.
Remember, it's all MCQ, Professor, right?
What?
It's all MCQ.
Yeah.
Multiple choices.
Yeah, MCQ says these are all MCQs. Maybe sometimes you have true and false. These are also.
All right. And you said this chapter you're gonna stop by the protein digestion.
No, yeah, after protein digestion, I'll stop it. Yeah.
Okay, thank you.
25 questions.
And once you take the quiz of this chapter,
Mhm.
in your next exam, it will not be in your exam. So we finish two chapters, and then you will have the second exam.
All right, thank you.
All right, thank you. I'll not take your attendance. If you want to stay for some other reasons, you can stay. Otherwise, you are free to go.
Thank you, Professor. Have a good day.
Have a good day, Professor.
Thanks, sir. Have a good day.
Same to you.